Good morning, dear student. How are you today? Hope you're fine and safe all the time. Today we are going to start in document 6, page 125. It's about antigen recognition by B lymphocyte. In the previous sessions, we noticed that the B lymphocyte, it's a type of immune cell or leukocyte that is produced and matured in the bone marrow. Because of that, it's named B lymphocyte, B related to the bone marrow. Antigen recognition is the cornerstone of a specific immunity. So our immunity, our specific immunity, depend on the antigen recognition. It's the first and the most important thing in the specific immunity. The body contains billions of different lymphocytes that are together able to recognize virtually any antigen present in nature, and thus provide protection against almost all potential invaders. So our body contains billions of lymphocytes. These lymphocytes are able to recognize, any antigen present in nature. And this antigen must be non-self antigen, to be fighted, to be catched or shooted by the B lymphocyte, However, how does a B lymphocyte recognize an antigen? Now we are going to start in part 1, antibodies, structure and classes. As you see, this is a B lymphocyte, and on the membrane of it, you will find many antibodies. These are called antibodies that are found at the surface of the B lymphocyte, and they are the main characteristics of the B lymphocyte. So let us notice what are the other characteristics of this antibody. So an antibody or immunoglobulin, antibody is also called immunoglobulin, is a membrane receptor displayed at the surface of a B lymphocyte only. So at the surface of B lymphocyte, you will find the antibody. What are the characteristics of this antibody? It is a Y-shaped structure. It's shaped like a Y letter, Consists of 4 polypeptide chain. You will find 2 heavy chain and 2 light chain, And also it includes a variable region and a constant region. What are the characteristics of the variable region? The variable region is characterized by the presence of antigen binding site. This is the variable region, This is the variable region. You will find it here also. That is found in the green or in a red color here. This is the variable region of the B lymphocyte, of the antibody. As you notice here, the variable region in red differ from this that is found in green or in blue. Is specific to a given antigen. Each antibody has 2 antigen binding sites. As you notice, 1, 2. Each antibody have 2 antigen binding sites. When I say antigen binding site, that means that the antigen will be catched by this binding site. Catched or shooted by. In the previous sessions, we noticed also that the antibody bind and neutralize the non-self antigen. Like here, as you see, this is a non-self antigen, bacteria. On the surface of this bacteria, you will find different type of antigen, and this is the antibody that is secreted by the B lymphocyte. The antibody have this binding site, the site that bind to the antigen of the non-self cell enter our body, and it neutralize its activity. Okay. Um, the variable region differ from one type of antibody to another. So each antibody have a specific variable region. As you notice here, this is an antibody, this is a B lymphocyte and this is a B lymphocyte. Each B lymphocyte have a variable region that differ from the other. Okay, as you notice here, it's like the triangle. It fit the triangular antigen, but here it fix or fit the circular antigen. Okay. Uh, However, we have also the constant region, a region that is constant. It's not variable for all antibodies. Okay, it's the same because it's constant. It's the same in all antibodies that belong to the same class. Okay, in fact, there are 5 classes of antibodies. We have class M, class D. So this is immunoglobulin M or antibody M, immunoglobulin D or antibody D, antibody G or immunoglobulin G, immunoglobulin A, and immunoglobulin E. So we have 5 classes of immunoglobulin: M, D, G, A, and E. Okay, this region is characterized at the bottom. This is the constant region, which is found at the bottom. Okay, the variable region found at the top of the antibody, while the constant region, this is the constant region that is found at the bottom. This is the constant region. This is the constant region of antibody. This is the constant region found at the bottom of the antibody, while this is the variable region. This is the variable region found at the top of the antibody. So this region is characterized at the bottom by a site used by antibody to fix itself on the certain membrane receptor. So the constant region is found at the bottom of the antibody, and it fixes itself on the membrane of the B lymphocyte. These are the main characteristics of the antibody, which are the main receptors of the B lymphocyte. Okay. Starting with the part 2 in this lesson, it's about specificity. But before, let us look at this document D. The one that is found in blue, this is the constant region of antibody. This is the constant region of the antibody, while the one that found in red, this is the variable region of antibody. Okay. Now, starting with the specificity. What do you mean by specificity? Antibodies are released, liberated from the surface of B lymphocyte to circulate in the body fluid, which is the blood. Okay, when a non-self antigen like bacteria, like a virus, or antigens other than the pathogenic antigens enter our body, especially circulated inside the blood, the B lymphocyte will differentiate into plasma cell, and this plasma cell will select this antigen and secrete antibody into the blood in order to bind and neutralize this non-self antigen. Okay, antibodies can recognize only extracellular. When I say extracellular, means that the antigen are found outside the cell. The antigen now does not infect the cell, does not enter into our self cell. Okay, they are still inside the blood. Okay, So they are called extracellular, outside. It's mean outside the self cell. Still have not infect the body cells, okay? When the antigen, for example, this bacteria, okay, this bacteria, when it enter our body and it is still inside the blood, okay, it does not infect the self cell of our body. It is still inside the blood, so our antibodies can recognize this bacteria or this antigen because it is still outside the cell. It's extracellular, so antibody can recognize only extracellular, still have not infect the body cell, okay? Because in the previous session we noticed that the T lymphocyte, not the B lymphocyte, can recognize the antigen that infect the body cell, that is intracellular, but the antibody can recognize only the extracellular, which means outside the cell, okay? Soluble, okay, which means dissolved inside our blood, okay? Free, not found in our cells, native antigens or membrane-bound antigen. When I say native antigen, it means only the antigen. When I say membrane-bound antigen, for example, this is a membrane-bound antigen. Antigen found on the membrane of the bacteria, okay? Native antigen mean antigen alone without fixing on the cell, on the membrane of the cell or the membrane of bacteria, okay? So when I say membrane-bound antigen, like in this way, these antigen are membrane-bound antigen, or as the antigen presented on the surface of RBC, antigen presented on the surface of red blood cells, okay? Because we know that there are A antigen or B antigen or both A B antigen found on the surface of the RBC. In case of incompatible blood transfusion, when the blood transfusion is incompatible, okay? The binding site of an antibody don't bind to an antigen as a whole, but rather to a small part of the antigen called antigenic determinant or motif or epitope, okay? So the antibody, this is the antibody, for example, this is the antibody does not bind to the whole bacteria, but it binds to this antigen, which is small in size, okay? So does not bind to antigen as a whole, not to the whole bacteria, but rather to a small part. This is the small part of the bacteria, which is the antigen found at the surface of this bacteria, okay? When I say bacteria, all this bacteria means antigen. Bacteria equal antigen. Bacteria means antigen, okay? And these are also called antigen bound to an antigen, to the bacteria, okay? So this antigen that is bound to the bacterium, this antigen or this, okay, as you see, they are called antigenic determinant, or they are called motif, okay, or they have another name, epitope, okay? An antibody can bind to a specific epitope since the antigen binding site of the antibody and the epitope have complementary structure like lock and a key. Okay, so this antibody, which have a circular binding site, can bind to a circular antigen. This antibody, which have a triangular binding site, can fix or fit on a triangular antigen. So they form as a lock and a key. Okay, an antigen may have a several epitope. As you see, bacteria, when I say bacteria, it's a non-self cell enter our body. I can say it's an antigen. Bacteria is an antigen. So an antigen means, for example, a bacteria may have a several epitope. As you see, this bacteria have epitope, triangular epitope, circular epitope, square epitope, or this this shape of epitope, may have several shapes of epitope and combined many different antibody. As you see, this is an antibody and this is an antibody, and these antibodies are different. They have different binding site. This is a circular and this is a triangular, as you see on the variable binding site. Okay, bind many different antibody. However, an antibody is not specific for an antigen, but rather for an epitope. So this circular binding site of the antibody cannot fix to the triangular binding site of the antibody or the square binding site of the antigen. Okay, so each antibody is not specific for all the antigen. Each antibody is specific for a specific antigen, but rather for an epitope, for a specific epitope. Okay? So a student say the bacteria means antigen. Any non-self cell or any non-self molecule may enter. Molecule means it's a protein enter our body. We can say it's an antigen. The structure that is bound on the membrane of this bacteria or antigen, we can say it's a small antigen bound to a big antigen, or we can say it's antigenic determinant motif or epitope. Okay, so each antibody bind to a specific epitope. So not all antibody bind to all epitope. Each antibody bind to a specific epitope. They form the lock and a key. Okay? Now the last idea in this lecture, it's about the cross reaction. As you see, this is an antigen. Antigen X. It's a non-self molecule enter the body, which must be binded which must be shooted or catched by this antibody. As you see, this antigen have many epitope. It have a circular epitope, it have a square epitope, and it have a triangular epitope. Okay, so an antigen may have a different epitope. As you see here in the bacterium, it have a different epitope. Triangular, circular, or square, or this structure of epitope. Okay, so this antibody, can one antibody bind different epitope? So can this antibody that have the variable site resemble the shape of a triangle, can it bind to the circular epitope or the square epitope? Of course, no. Okay, it can bind only to the triangular epitope that resemble its shape. That resemble its shape, that makes, okay, look like key. Lock and the key. Okay, so an antibody can bind to a specific epitope since the antigen binding site of the antibody and the epitope have a complementary structure. Okay, so this binding site of an antibody is complementary. Okay, can fix or can fit the triangular epitope only. Okay, this is called a cross reaction, when an antibody bind to different antigen that share same antigenic determinant. Okay, cross reaction when an antibody bind to different antigens that share same antigenic determinant. As you see here, this is antigen X and this is antigen Y. These two antigens are different. If you look at the structure, this is circular and this have this shape of epitope, so they are different antigen. This is antigen X differ from antigen Y, and this is one antibody. So an antibody, okay, one antibody can bind different antigen. two different antigen. This is two different antigen, antigen Y and antigen X, okay, that share same antigenic determinant. And they have the same antigenic determinant. They have the same epitope, triangular epitope here and a triangular epitope here that can be fixed by the triangular binding site of the antibody because they are complementary and they look like the lock and the key. Okay? What do you mean by immune complex? A molecular aggregate formed by binding of several antigen to their specific antibodies. This is called an immune complex, as you see here. Okay, so this is a cross reaction. This is the cross reaction because antibody bind different antigens. Okay. When I say the immune complex, it means a molecular aggregate يعني هجوم of the antibody by binding of several antigen to their binding to several antigen. So the antibody binds several antigen. It means that the antibody fight by forming a molecular aggregate against this non-self antigen. And this reaction takes place only outside the cell, extracellular, as I told you, extracellular. So this antigen is still not found, not infect our cells. This was all about document 6. Hope you understand it very well. If you have any questions related to it, please ask me. Okay?